for antibiotic use in the field, in other words, for a patient, is on the insert. The FDA requires that whenever you purchase a box of antibiotics, there's an insert in it telling you allergic reactions and all of those things. It also says the width of the zone of inhibition considered by the FDA to be sensitive. Now, if you take the most commonly used antibiotics and average them all together, you will get 21 millimeters. So remember, greater or equal to 21 millimeters is what we use as sensitive in our class. Greater or equal, now we're talking about the diameter, so you can measure from the center of the disc to the nearest growth times 2. Diameter equal radius times 2. Because some of these intersect, and it's very hard to measure their diameter. So again, you take your ruler and you measure to the nearest growth. Now, when you're recording on this, I'll get to you. When you're recording the information on your antibiotic disc, remember that if you see little dots next to the disc, that whole thing is garbage. No longer can be used because that is a mutant. So if you looked at this picture here, you will see that that whole zone around ampicillin is not completely clear. It's slightly cloudy. That means it's filled with mutants. That means in the population of the bacteria you put on that plate, I mean, you can't see it as well as you should. In the population of bacteria that you put on this plate, there are significant numbers that already have resistance to that antibiotic. Therefore, that antibiotic is considered useless. You would only use one that has a completely clear, without any dots in it, region or zone of inhibition. So remember, one, you can't look at ones that are slightly cloudy. That's garbage. Those are mutants. You have to look at one that has completely clear, and it has to be at least how many millimeters? 21.
21.
What?
That's less than.
Oh, thank you.
You're welcome.
All right. Finally, you need to record what's called synergism. One of the amazing things that we've discovered with antibiotics is that if you use a certain two antibiotics together, they have a greater effect than either one of them separately added together. And it's called synergism. There's a greater effect than an additive effect. So, how do you determine synergism? When two zones of inhibition intersect, like these two did, without an indentation coming between them, that's synergism. So together, they have this zone of inhibition when used together. These two have that zone of inhibition when used together. This one. But these two have no synergism because, see, the growth comes between them. So you have to record zones of inhibition, and remember, you can write just sensitive or resistant, and if it's less than 21, you don't have to, you just write less than 21 ampicillin. No good, resistant. But on these, you have to write what they are individually and what they are synergistically. So anyone have trouble reading an antibiotic plate? Oh, you just measure in millimeters the width of the zone of inhibition, the diameter. And the best thing to do is just measure from the center of the disc to the nearest growth times 2. That will give you a better than trying to get what's the right. Okay. Anyone else have trouble with any card? Any card that you had difficulty understanding what to do? Let's see. All right, let's do some unknown problems. Yeah.
For the colony morphology, when you talk about the different descriptions about where can we find that?
Yes, okay. I'll show you. Colony morphology descriptions. Right here, colony morphology.
When you say like church dome, because I remember on the last test you described so many different ways.
Right. Fulminate is a church dome and uvinate is like a nipple. Also, a couple of you have Serratia marcescens, and it says on here that it grows pink at 25 and white at 37. And I kept telling people, go to colony morphology and read that. They kept saying, no, mine's contaminated. It's got both pink and white colonies. So, this tells you the colony shape, punctiform, circular, filamentous, irregular. I only make 2 of those questions, so it's not really that significant. Probably will take it out because I want to add more on the black cotton arm. Okay, let's do a couple of these. Everyone did see this word question yet? See if I can find one. Oh, the screen. Yeah, I did. anybody know of one of these, which one of these links has a midterm question on it?
And then there's one, it grows on the table. It's a robot, right? And if it grows in the jar. We're gonna meet tomorrow.
Yeah. Well, anybody have a word question that they have trouble with, and we'll do it on the board. Okay. Yeah, this was good. All right. I'll read it out, and then we'll do it. This bacteria grew on salt agar but showed no trace of calcium ion.
What do you say? This bacteria on salt agar didn't show us.
Or let's do this, because that one's going to be easy. This unknown rod was recovered from a water sample. It stained blue on the stain for lipids in the cell wall area and pink on the gram and stain detect survival structure. So this is 202 and it's spring 2013. And it was from a rod, from a water sample, and it's gram negative, spore negative, and acid fast negative. Okay, it grew nice round raised dot on nutrient agar and showed no clearing on the plate that looks like a frosted bathroom window. That's the lipase or spirit glue test. So, showed no clearing, meaning it was negative. It showed no halo on the stain using India ink, so that's the negative stain, negative. There are cheers stacked up there, and you'll have to grab one. The lysine slant showed nothing. Lysine slant is lysine iron, so that means most probably the H2S is negative. The hanging drop showed shadowy microbes but no tumbling after 20 minutes of focusing under oil. Well, that doesn't matter. So you don't have any results on that because 20 minutes, you have to do it in 5 to 15. Oxygen showed a tampon string hanging from a thick growth at the top of the tube extending to the bottom, facultative arrow. There was a solid red line down the center of the gel stab, so now we know our motility is negative. EMB showed a deep chunk in the agar where it was inoculated, but no growth, as if the loop was too hot during inoculation. So I don't think that's any information. I think that's just bull. McConkey's was broken in the incubator, damn. And the lazy mutant test was white. So what's your lactose? ONPG was white. What? It's negative. Okay, the nitratase test was pink after reagents and zinc. Yep. This microbe showed bubbles on a clean slide test, and that's the catalase positive. Dioxic growth was not done, so that's the cliggers. Green agar had growth that was the color of the media, So that means it is lactose negative. We already knew that. With some dark crystals, but no change in the color of the agar. So the dark crystals might indicate hydrogen sulfide, but again, not a real definitive answer. Let's see, we do know that it was, since they drew on it, it is gram negative. We already knew that. Okay, let's see. Urea broth was yellow, so the urea test is negative. And as was the dextrose, so now we know our glucose is positive. Dextrose is another word for glucose. Mixed acids test was yellow, so MR is negative. Litmus milk was purple without any waves, so casein is negative. And purple is another lactose, negative. Remember, the clear liquid above a cottage cheese curd is called whey. Remember, she was eating her curds in whey? You don't remember that little nursery rhyme? So sad. All right, the old test with the oxidase reagent was gray, so oxidase is negative. The broth where you took 2 and incubated and inoculated 1 for 2 weeks. This one for 2 weeks was beige after reagent, so that's BP. And showed nothing after sitting overnight. Of the carbo broth, 2 were yellow and 1 was pink. So that means glucose, we know was yellow. We know that lactose was negative, so that means that mannitol was positive. Cow horns was removed from eating cool. What's it? Was a gel, so gelatin is negative. The tryptophanase, which is the endo test, was green only at the top, so that is endo negative. The clear broth showed bubbles in some solids, so citrate is positive. What's the genus? See if we can figure it out. All right, so we have a glucose positive, we have a citrate positive. Our hydrogen sulfide is confusing. So you're going to have to answer this one. If hydrogen sulfide is positive, the answer is blah. If hydrogen sulfide is negative, it's this. Because this test indicated it might be negative, but this one indicated it might be positive. So you'll first go down saying it's positive. Our indole was negative, our gelatin was negative, our mannitol was fermented, and our lactose was not. So if H2S is positive, the answer is salmonella. Now let's say if H2S is negative. If it's negative, what's our BP is negative? It's Citrobacter proteus. Problem? Want me to read it out again? Yeah.
Random question. For the Kia test, like, you say that Kia is yellow. What do you mean by yellow? Is it the blood or the swab?
No, you have to use another test to figure out where the yellow was, whether it was both or in one spot.
Okay.
Are we going to have like choices or do we write down?
No, you won't have choices.
Okay, so we write down.
Yeah, you would write this down. And believe it or not, this would give you 30 points. Because each one's 10. So Citrobacter is 10, Proteus is 10, and Salmonella is 10. So that'd be 30. If you only wrote this one, you'd only get 10. That was a really good one. I like that one. I think I like that. Yeah, this is the last one. Shall I do another one?
Yeah.
Yeah.
Say that was 202?
It was 202 on the spring 2013 EXLPH.
I found it on the website.
Huh?
I found it on.
Yeah, it's on the website somewhere. You know where it's at?
Yeah, I just found it.
Oh, good.
Let me see if I can find it on yours.
Yeah, would you?
Yeah.
That way we can put it up on the overhead and people can see it. Okay, I'm gonna try to do number 203 then. Okay, so on 203, I'll try reading it and she's gonna try finding it. The unknown microbe was a coccobacillus. It took the second stain on all the differential stains. So if it took the second stain on all the differential stains, on the gram stain, the first one you put on is crystal violet, the second one you put on is saffron, so it's gram negative. And if it took the blue on the acid fast, it's acid fast negative. And if it took the green on the endospore, you're not really sure because, you know, it can show green on the endospore, so it might be positive, but remember it's gram negative, so probably not. If it took green on the spore stain, it's probably just garbage. Remember, lots of things take the green on the endospore stain. Okay, the test where you took 2 tubes of it and incubated one for 14 days was brown after reagents. So that would be the VP. And brown is negative. Did you find it?
Yeah.
Yay! We're doing this one, and I'll turn off down the lights so that
Red
this
is
one goes
positive.
there. So we're doing this one. The test where it took 2 tubes and incubated one for 14 days was brown after reagents. Hydrogen peroxide on the test did not show dissolving. So did not show dissolving would be its catalase positive. Remember, when it looks like sugar dissolved in water, it's negative. When it bubbles, it's positive. So if it didn't show dissolving, it did show bubbles. I know, screwy. 2 of the 3 PR broths were orange. Glucose, lactose, mannitol. So 2 were orange, which means negative. The green agar remained the same as inoculated, and the agar plate that uses neutral red showed clear growth. So neutral red is McConkey's, and clear growth is lactose negative. So we now know one of those is negative. The solid red down the center of the gel stab means motility negative. The broth that has an upside down little tube in it was gray or gray beige after reagents A and B, but was red after fairy dust. So it's nitrate negative. It showed blue on the stains that detects lipid, but it was just a shadow on the stain that uses India ink. So the negative stain is negative. And the spore stain, the acid fast stain is negative. Just a shadow on hanging drop means no motility, we already got that. The contaminated water test was -1, +1, -2. So that's indole, methyl red, BP, citrate. -2? -1, +1, -2. -1, +1. Okay. Yeah, too many numbers there. that's right, okay. And that supports that. So this gives us an MR, gives us a citrate, and it gives us an indole. All right, blah, 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 blah. One milk test was cloudy and a colony appeared round. So, yeah.
MR.
MR is positive.
MR?
You said 1 + 1?
Yeah.
How did you remember? Plus
Oh,
one.
yeah. Plus one, IM, yeah.
Remember? Yeah.
All right, let's see, the
This one is the cake.
cloudy at the, where was I? One milk test was cloudy. So cloudy means the casein is negative. Brown and white, microbes grew cream and raised no longer. Cloudy down to the top tube with a whiz down the side. So we have what on the broth that's very fragile. So again, it's facultative. Urea showed yellow and after 5 days. That doesn't give you a lot of information because urea is supposed to be red at 48 hours. So you put a parentheses that you're not confident in that result. Gray then black, so oxidase was negative. The dioxic growth had 2 colors, while the test that so the dioxic growth had 2 colors, which means that the glucose was positive. That means that the mannitol was negative, and the hydrogen sulfide was negative. 2 colors, not 3. Okay, so let's see what it's doing here. Horse's hoof was solid, so that's gelatin. Amylase was negative, so now we're done. All right, let's see if we can figure her out. We have a glucose positive rod that's gram negative. Citrate was negative. Hydrogen sulfide was negative. MR positive. VP. So lactose not fermented means Shigella. Non-modal. Questions?
How did you decide it was mannitol after the mannitol positive?
Oh, because it said the dioxic growth showed 2 colors. So the 2 colors that we already knew that the lactose was negative, so that's red is 1 color. And the other color would be, the other one had to be, there's only 2. So there's red and yellow, so this one had to be positive. And we'd already found the hydrogen sulfide from another test. It didn't show up on the green auger, so it was negative.
No, because after you read that part, you said that means glucose is positive and mannitol is negative. So I just did-
Right, because it said 2 of the 3 are orange. So if this one's negative and that one's positive, this has to be the other negative. Okay, so now I want to take, do you want to do another one?
Yes.
Okay, let's do another one. Do we want to do loose stools? Okay. We'll do 204 and then we'll take multiple choice questions. Where was it what?
This is all H, right?
She knows.
I can show you.
All right, so 204, this one was taken from a loose stool movement. What did I tell you in class that means? Gram-negative rod. That's what poop is. All right, it didn't grow on the salt plate. Well, golly, we know now it's gram-negative twice. It's stained blue on the stain for lipids in the cell wall area, so that's acid-fast negative. Solid pink was slightly longer than wide shape, so we know it's again a rod. And And it's pink, so it's 4 negative. In Thio, it was heavily cloudy at the top with a wisp down to the bottom, again, a facultative. And the test to detect impure water was +1, -2, +1. So that's indole positive, MR negative, BP negative, and citrate positive. The O2 test was gray then black, so that's oxidase negative. On the iron slant deep, there were 3 colors. So 3 colors means that we have yellow, red, and black. So we now know that our hydrogen sulfide is positive. Now we need something to tell us which was the lactose and which was the glucose. The 12-hour wet mount showed vibrating structures. So vibrating structures means motility is negative. Litmus milk had purple and whey. If it has whey, it's casein positive because that's the clear liquid above a curd. And purple tells us that the lactose was negative, which means the glucose was positive. Thick red line down the gel, another motility negative. Gelatinase was noted, so it's gelatin positive. And the carb broths were +1, -2, so that would be glucose, lactose, and mannitol, +1. And that gives us that gives us that, and that gives us that. All right, so let's now check it out. Glucose positive. We have a citrate positive, hydrogen sulfide positive, our indole was positive, our gelatin was positive, our mannitol was not fermented, Proteus. Okie dokie, multiple choice questions.
I have a quick question for the wet mount. You said it was negative because it also says it's negative because it's in 12 hours.
Right.
So, because vibrating is negative already, but it's 12 hours, you've already just.
Yeah, you would not know what your result was, so your gelatin stab would confirm that it was negative. Because it's a straight line. Other questions?
You know how you said the average for the midterm is 50%? You have like a couple of days sometimes?
No, I have, she wants to know what the normal grade pattern is on the midterm, and the normal grade pattern is 40% Fs and about 55% flat out A's. People mean A or F.
Wow.
They know the word stuff, and they balance out what they miss on the multiple choice by making huge numbers of points with the word questions and ace it. And then there are those that didn't bother to learn the word questions that don't know the cards, and so they flunk. Yeah.
For the first word problem that we did, is the reason why we did the if-then statement for the hydrogen sulfide because there was 2 conflicting tests. So
Right.
one said crystals and the other one, it wasn't sure
Right.
if-then.
Exactly. She wanted to know why on the first word problem I put if the hydrogen sulfide is positive, it's this answer, and if it's negative, it's this answer, because in the reading, it gave us 2 conflicting results that we couldn't conclude which one the hydrogen sulfide was. It said we saw crystals on a plate, on a Hectoan plate. Well, crystals usually is hydrogen sulfide. And on the other one, it said there was only no color in the Kliger's iron. And no color at all would indicate that it was red with no black, so it would give us a negative. So we had both a positive hint and a negative hint, and there was nothing to decide between. So we had to answer 2 different ways. And you know, that's good. Other questions? Anyone want to read a multiple choice question you're confused with? Yes?
Like, the top person gave us last week for number 39. It was kind of like a weird question. Narrated about
Yes, can I see it?
Yeah.
I'll read it out. It may be one of those.
39.
There was one on there that I just, number 39. Okay. Cligger's iron that is properly inoculated and incubated and has a red slant with a red-black butt on the of the auger indicates that what test should be what. Okay, a red slant means that all the lactose tests should be negative. So that would be hectoan auger growth with dark crystals. Well, growth means it's gram negative. And the red-black, so the black would be the crystals, so that one would be right. EMB growth is clear, is lactose negative, and that agrees with the red slant. Phenol red lactose broth pink agrees with the red slant. Phenol red dextrose orange agrees with the red bud.
14 and 9.
And so none of these is incorrect as right.
For it to be having the profile has to be
Can be independent. Anyone else have a multiple choice question? Do you want to go over the hints? Somebody have something that they want reviewed? Lack, trip, what?
The stages of glycolysis.
Okay, the
Can we go
different
over it?
kinds that exist?
No, there are the stages for glycolysis, right? Like first stage, second stage, or third stage.
Different stages.
Stages? No.
Where's the question?
Um,
I mean, you know, all living things except for one go through glycolysis. They break down glucose to 2-pyruvate. 80% of them at the same time of bacteria also go through this pentose phosphate shunt, where they take 5 carbon sugars and break them down to pyruvate. And one goes through that interdeuterol pathway, but it still gives you pyruvate. So, is that what you mean?
Yeah.
Okay. So, if you recall, on glycolysis, we said it was a universal respiratory pathway of all living things, and all living things did it first in the cytoplasm. So, glucose broken down to 2-pyruvate.
Here we are.
And this happens in the cytoplasm of all living things. And it's called anaerobic respiration, specifically glucose splitting glycolysis. And you have to add in 2 ATP, 4 ATP, and you get given off 2 ATP. And 39 genera of bacteria do this. One does an alternative pathway called interdeuterol, which I cannot spell. G-O-U-T-E-R-O-F-F, I think. Who knows? Interdeuterol, and it does not go that way, but it does give you 2 ATP, and it does give you 2-pyruvate. Now, remember, one genus does this. Everything else does the Embden-Meyerhof glycolysis pathway. And of the 39 genera that do this, 35 at the same time are running the pentose phosphate shunt. And they're still getting 2 ATP, and they're still getting 2 pyruvate, but they start with a 5 carbon sugar, and they're running it at the same time they run the 6 carbon. So all of this happens in the cytoplasm. 99.9% of all life does glycolysis, the Embden-Meyerhoff pathway, anaerobic respiration, glycolysis, sugar splitting, where you get 2 ATP and 2 pyruvate by adding an ATP to each end and splitting it in the middle. One genera of bacteria does a different method of splitting glucose called interdeuteroff pathway, but the good news is it still gets 2 pyruvate and 2 ATP. So you still end up at the same place. And what comes after this? With most bacteria, the facultatives and the anaerobes, what happens after pyruvate?
Fermentation.
Fermentation. And that also is in the cytoplasm. And is also anaerobic. And remember, we have 11 pathways of bacterial fermentation. BP, which is the 2,3-butadiol, your BP test, which is Enterobacter, mixed acid fermentation pathway, which is E. coli, butyric pathway, which is Clostridium, and so forth. So, anyone have any particular multiple choice questions? Yeah.
The electron transport takes place in the cell membranes and generates ATP according to what Nobel Prize winning theory?
Chemiosmosis. Yes.
164.
You'll have to speak up louder, I can't hear
A
you.
growth medium consisting of only 50 milliliters of water, 1 liter of agar, and a mole of NaCl would be considered A synthetic, B complex, C defined.
Define.
What
When
number
you
is
have
that?
a molar recipe of something, it's called defined media. What number was it?
106.
106?
Can you repeat that? When you have a mole. So anytime it says, even though it says like 50 milliliters of water or 1 liter of agar, it's the standard to all.
Yes. 1 liter of agar is standard to any agar recipe, and you don't have to have, because agar is a, I guess, organic compound.
And then the question before that, it says it has 2 pounds of roasted soybeans and 1 mole.
Ah, yeah. And what?
And 1 mole of calcium chloride and specific gram amounts of vitamins would be considered defined synthetic medium, complex medium, chemically non-defined medium, and both defined and complex or enrichment.
Okay, this one would be just complex, because the one item you put, what was the first one?
It says-
Ground soybeans.
Yeah.
Ground soybeans is an off-the-shelf item, and anytime you put an off-the-shelf where you don't have a definition of what it is, like oatmeal, then that is a complex agar. Okay, defined as a recipe with molar quantities of everything, every element. Okay, yeah,
53 on test R.
okay.
It says organisms that use carbon dioxide to synthesize organic molecules are called A, autotrophs,
Yes.
saprophytes, heterotrophs.
Autotroph.
It's autotrophs. So if it's exactly photoautotrophs, but if autotrophs is there, that could be the answer.
Yes.
Okay.
Question?
This isn't multiple choice specific, but for the trip operon, can you go through the different types of repression on where it binds? Because I think your explanation.
Are you talking about attenuation?
Yeah, so when there's one where like
Okay,
it
on the
binds-
trip operon can be controlled 2 main ways. On the trip operon, if the excess tryptophan binds to the repressor protein as a co-repressor and sticks to the operator, then that is called repression. If the excess tryptophan sticks to the fourth, 2 places on the third, or one place on the second of the 5 intermediate enzymes in the pathway, it's called attenuation. If by chance, now this doesn't happen, but if by chance the product sticks to the first enzyme's allosteric site, that's feedback. So remember, if you have these enzymes, and then you get a product. If the product sticks to the first allosteric site, that's feedback. If it sticks to some intermediate enzyme in the pathway, that's attenuation. And of course, if it sticks to a co-repressor, remember, tryptophan stuck to the co-repressor and stopping it at the operon, the operator here, by matching to the operator, that was repression. Other things? Yes?
So which one is the least efficient indirect method of attack?
Well, they were all in order, so on direct methods, the most efficient is feedback. The second most efficient would be competitive inhibition, and you just move up that list. Remember, feedback and competitive inhibition are all direct controls, not indirect.
It says which of these fermentation pathways do not produce gas? And it's
Only lactic acid doesn't produce carbon dioxide. Is it in there?
homolactic?
Yeah, that's homolactic. I'm not going to get that critical in this one. That particular term, I put a lot of emphasis on memorizing each one of those 11 pathways from pyruvate. I'm only going to go over the one that leads to your methyl red test and your VP test, or your test. I'm not going to ask that you memorize all the end products of the different fermentation pathways this time. We didn't have time to emphasize it as much as I usually do. Other questions?
Which of the following is an example of vertical gene transfer? Conjugation, transposition, electroporation, progeny, and transduction.
Progeny. You want to know which of the following is an example of vertical gene transfer? When you're passing from mother to child, that's vertical. Conjugation, transduction, all of those are horizontal. Translocation is even horizontal. Other questions? Are we giving up? I'm going to move into lab if you don't have any other questions.
One more.
She has one.
For Simmons, the reactions produce ammonia.
Yes, Simmons, the reactions produce ammonia, but again, I'm not getting into that much detail this time. Good for you. No, you know it already, so it's not. You know, there are several ammonia tests. That used to be a question. How many of our tests going through the process is ammonia, one of the end products? Well, citrate was one, urea is one, and the nitrate test is another test that ammonia can be an end product. But I'm not asking that question this time. It's too complicated. Keeping it light. Hitting the word problems this time. Each term I emphasize something a little bit.
You have to like outline the lac operon.
Outline the lac operon? No, just to be able to answer multiple choice questions on it. Yeah, no short answer this midterm. Don't have time. Don't have time to grade them. Don't have time for you to write them. Yay! Other questions?
Yes.
Yes.
Okay, a microbiologist is studying the behavior of a hydrolytic enzyme found in alcohol. This enzyme is active because of the lactose at 35 degrees Celsius and pH 2.0.
She's reading the question that has to do with how do you turn off an enzymatic
Is
system.
it to raise the
temperature.
Raise
by
the temperature 2 to 3 degrees
So
centigrade.
it's adjusting the medium to 39 degrees Celsius,
Yeah,
right?
whatever from the optimum temperature, raise it 2 to 3 degrees centigrade, you shut off the system. And then I'll have a whole bunch of misleading crap in there. That's the only thing that's important,
Why?
just to confuse you, to make you happy. Yeah. Oh, Jesus, Mother of Heaven. Go to the video on it and look at it because it is lengthy. Yeah, but there is a video, do chapter 5, 7 video, and you may, there's like 4 of them. You'll have to find the one that has the discussion. Other questions?
Yeah, Professor?
Yes.
One of my questions, which is the least efficient direct method of control?
The least efficient method, which one is it? 48. Among the ones, those listed, which is the least efficient direct method? Feedback is the most efficient of the direct. Enzyme repression is indirect. Attenuation is indirect. Enzyme induction is indirect. Catabolic repression is indirect. So since the only one there that's direct is feedback, that's it.
So least efficient.
Of the direct. Yeah, well, of the list. Okay, so you may go unless you're staying for lab. If you're staying for lab or confirming, you can confirm your genus and stuff like that. Anything else that you need, you can contact me on Google Plus on Sunday. If you don't have your genus figured out, but you figure it out over the weekend, email. Anyone have any other questions? Could you hit that off?